Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a future where doctors can look at your individual ovarian cancer tumor and know right from the start which treatment is most likely to work.
Yeah,
That kind of personalized approach could mean better outcomes and, less time spent on therapies that just aren't effective.
exactly.
So this deep dive explores a really interesting discovery that's bringing us a step closer to that reality.
We're going to be looking at a specific gene. It's called NFX1 type zinc finger, containing one bit of a mouthful. So we'll just call it ZNFX1 going forward.
It's okay, ZNFX1.
And this gene seems to play a really crucial role in how some ovarian cancers respond to treatment, especially those that have, well, unfortunately become resistant to the initial therapies.
Right, the therapy-resistant ones. So our understanding today comes from a study that was just published in Cancer Research, a pretty major journal.
Yes, and there's also a helpful article from the University of Maryland School of Medicine that kind of breaks down the science for us, which is useful.
Definitely. So our mission for this deep dive is pretty clear. Understand what this ZNFX1 gene actually does, how scientists figured out it was important in ovarian cancer, and crucially, why this might be such a big deal for future treatments.
Right. So the big question really is, what exactly did these researchers do to uncover the importance of ZNFX1? How did they find this link?
Okay, yeah, let's get into the study itself. What was the main thing the researchers were trying to figure out? What was their goal?
Well, the central aim was to identify factors,
Yeah.
things they could measure, that could predict how patients with advanced stage ovarian cancer would respond to certain cancer treatments.
So looking for predictors, how they do that?
They put together a team from various institutions, and they analyzed existing collections of data from ovarian cancer patients. Think of it like sifting through this huge library of information, looking for patterns that kept popping up.
So they weren't starting from scratch. They were looking for clues within this mountain of existing patient data.
Precisely. And by examining these databases, the researchers found a pretty significant link. They noticed that when patients had high levels of this ZNFX1 gene in their cancer cells, it often correlated with a better response to certain therapies.
Oh.
This suggested that, the amount of ZNFX1 present could potentially act as a sort of signpost indicating how well a treatment might work for that patient.
So simply put, higher levels of ZNFX1 might be a good sign for patients on certain treatments. That sounds like a potentially huge insight for dieting therapy.
It really could be.
Now, building on that pattern they saw in the data, did they see something similar when they looked at actual patient outcomes, maybe in a clinical trial?
Yes, they did. They found a similar connection in a phase 3 clinical trial, which is a pretty advanced stage of testing. This trial involved patients whose ovarian cancer had unfortunately stopped responding to their initial treatments, the therapy-resistant group again. These patients were receiving a combination, the drug, bevacizumab, along with standard chemotherapy. And the study found that higher levels of ZNFX1 were associated with improved overall survival in this specific group.
Wow. Okay, even in that difficult-to-treat group.
Exactly. The fact that ZNFX1 levels seem to point towards a better outcome there is really quite significant.
And it wasn't just about seeing this link, was it? I think the study also looked into what might be controlling the ZNFX1 gene itself.
That's correct. They wanted to understand what could be affecting the activity, the expression of ZNFX1. And they discovered that two types of existing cancer drugs, these are called DNA methyltransferase inhibitors and PARP inhibitors, could actually increase the expression of the ZNFX1 gene.
Oh, interesting. So existing drugs might boost this beneficial gene?
Yes. This is a key finding because it suggests that these drugs might be working, well, at least partly, by boosting the levels of this potentially helpful gene.
So these drugs aren't just like directly hitting the cancer cells. They might also be flipping a switch on a gene that helps fight the cancer itself. That leads to the question, what does the ZNFX1 gene actually do inside the cancer cells?
Right, the mechanism. The research delved into that as well. They found that ZNFX1 plays a role in something called a pathogen mimicry response. And this happens within the mitochondria, the little energy factories inside our cells.
Pathogen mimicry, that sounds like the cancer cells are pretending to be invaded by something, like a virus or bacteria.
In essence, yeah, that's a good way to put it. Think of it like the cancer cell is accidentally waving a red flag, making the immune system think it's under attack. This response within the mitochondria then acts as a trigger for tumor-suppressing inflammatory signals.
Okay.
And ZNFX1 appears to be a key director, a master regulator of this internal alarm system.
So ZNFX1 is actually helping the body's own defense mechanisms kick in, but from within the cancer cells themselves.
That's the picture that's emerging, yes. And to investigate this further, the researchers did experiments in the lab. They basically removed ZNFX1 from ovarian cancer cells.
Okay, took it out. And what happened?
What they observed was quite revealing. These cells, the ones without ZNFX1, started to grow more quickly, and they were more prone to forming tumor-like structures, these sort of clumps, in the lab dishes.
Ah, so removing it made the cancer act more aggressively.
It strongly suggests that ZNFX1 normally has a tumor-suppressing effect. Taking it away lets the bad behavior run wild, essentially.
That really highlights its importance. The study also used some pretty sophisticated techniques, right? Something about reading the genetic messages inside the cells.
Yes, exactly. They used ribonucleic acid sequencing. It's a powerful way to see which genes are active and what messages they're sending out inside the cells. This helped them explore the downstream effects of ZNFX1.
And what did that show?
This analysis revealed a connection between the inflammasome signaling, that internal alarm system we talked about, the one ZNFX1 regulates, and the formation of abnormal blood vessels within tumors. Angiogenesis, basically.
Angiogenesis, right. We know tumors need a good blood supply to grow and spread, don't they? So if ZNFX1 influences that.
Exactly. Tumors are greedy. They need those blood vessels to bring them nutrients and oxygen. The fact that ZNFX1 seems to impact this critical process further underscores its role in controlling cancer development and growth. So putting all these pieces together, the patient data, the clinical trial results, the lab experiments, the genetic sequencing of the study, concluded that ZNFX1 acts as a tumor suppressor. It does this by orchestrating this pathogen mimicry response in the mitochondria, which in turn affects tumor growth and blood vessel formation.
Okay, so we understand the what of the study, the findings. Now let's pivot to the so what. What does this actually mean for the future of treatment? What are the real-world implications?
Right, the implications. Well, one of the most promising possibilities is that ZNFX1 could serve as a biomarker, a predictive biomarker.
A biomarker, meaning?
Meaning, in the future, doctors might be able to test the levels of ZNFX1 in a patient's ovarian tumor biopsy.
Yeah.
Based on those levels, whether they're high or low, they could potentially predict how likely that patient is to respond well to certain treatments, like maybe those involving bevacizumab, as seen in the trial.
So it could help personalize treatment decisions.
Exactly. It could really change how we approach those decisions. Instead of maybe a trial and error approach sometimes, we might have a clearer indication right up front about what's most likely to work for that specific individual. More personalized medicine.
And the fact that those existing drugs, the DNA methylthingy inhibitors and PRP inhibitors.
DNA methyltransferase inhibitor.
Right, those. The fact that they seem to influence ZNFX1 levels is also a really significant point, isn't it?
Absolutely. That finding offers a potential explanation, a mechanism for why these drugs might be effective in treating ovarian cancer, and maybe other cancers too. We didn't fully know how they worked in this context. And it also opens up exciting possibilities for exploring combination therapies. For example, could we combine these drugs, maybe even with other treatments, in a way that specifically maximizes the boosting of ZNFX1? That could potentially lead to even better patient outcomes.
So we could potentially fine-tune how we use these existing drugs specifically to leverage the power of ZNFX1. That's really interesting, but like with most research, there must be limitations, right? Things we still need to figure out.
Oh, definitely. You're right. It's not the complete picture yet by any means. Well, the link between ZNFX1 levels and treatment response is certainly encouraging, especially seeing it in both the big data collections and a clinical trial. More research is absolutely needed.
Like what kind of research?
Well, we need to definitively establish ZNFX1 as a reliable predictive biomarker. This will require further validation in larger prospective clinical trials. These are studies designed specifically to test its predictive power going forward, not just looking back at old data. So the initial signs are good, but we need more robust evidence.
That makes sense. Need to be sure it really works consistently. And what about understanding how ZNFX1 works inside the body, not just in lab dishes?
That's another key point. While the study gave us valuable insights into how ZNFX1 functions in those controlled laboratory settings, we need more research to fully grasp its complex role within the actual environment of human tumors in the body.
Because it's more complicated in a real person.
Exactly. The tumor microenvironment is incredibly complex. There are immune cells, structural cells, blood vessels all interacting. What happens in a dish might not perfectly mirror what occurs in a patient. We need to understand ZNFX1's function in that real-world context.
Got it. And those two types of drugs that can increase ZNFX1, the DNA methyltransferase inhibitors and PRP inhibitors, what are the limitations or next steps there?
Well, those findings are promising, definitely. But we still need to figure out the optimal way to use their ability to boost ZNFX1 expression in actual treatment strategies.
How so?
Questions remain, like what's the ideal dosage? When should these drugs be given? What are the best combinations with other therapies to really maximize this ZNFX1 boosting effect for the benefit of patients? So while we know these drugs can boost ZFX1, we still need to learn the best way to harness that knowledge clinically.
Right, translating the lab finding into practical treatment plans. Now, it's always valuable to hear what the experts who were actually involved in the research think. What did they have to say about the significance of this discovery?
Yeah, so Dr. Fairuzvi Rasool, who is a senior author on the study, really emphasized the potential for personalization. He stated that ZNFX1 may help in delivering more personalized therapy to patients with ovarian cancer, essentially suggesting that understanding a patient's ZNFX1 levels could allow doctors to tailor their treatment more effectively.
That really reinforces that potential as a predictive tool, that biomarker idea. Were there other expert perspectives mentioned?
Yes, Dr. Tafike Owonikoko, who is the executive director over at the University of Maryland Marlene and Stuart Greenbaum Comprehensive Cancer Center, he also highlighted the significance of identifying ZNFX1. He noted that this discovery really paves the way for more research in a clinical setting. And he mentioned it aligns with their institution's commitment to advancing new therapies for patients who are facing this very challenging disease. So real enthusiasm about the next steps.
It sounds like the experts are definitely optimistic about the future research this discovery opens up. Okay, so let's try and wrap this up. What's the big takeaway here from our deep dive today?
I think in essence, we've explored the really exciting discovery that higher levels of this gene, ZNFX1, in ovarian cancer cells seem to be linked to better responses to certain treatments. This is especially notable in cancers that have become resistant to other therapies.
Right.
We also learned that some existing cancer drugs, the DNA methyltransferase inhibitors and PRP inhibitors, can actually increase the levels of this potentially beneficial gene. And crucially, that ZNFX1 appears to play a key role in triggering an internal pathogen mimicry response within cancer cells, an alarm system that can help suppress tumor growth and maybe even affect blood vessel formation.
This really does feel like a significant step forward in, peeling back the layers of ovarian cancer and figuring out how we might be able to treat it more effectively, more personally in the future.
Absolutely. There's still work to do, as we discussed, with validation and fully understanding the mechanisms in patients,
Of course.
but it's a very promising lead.
Definitely. For those listening who maybe want to learn more, the original study itself is available in the journal Cancer Research, and the University of Maryland School of Medicine also has that helpful article summarizing the findings. So we definitely encourage you to check those out if you're interested in digging even deeper.
Yeah, good resources.
So that brings us to the end of this deep dive. Thanks for joining us.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.